Hello, everyone. Thanks, Sean, for the introduction and the nice words from Sean and Derek. And especially fun to hear John Brady, who I'd not met before. And his description of SGLT2 inhibitors was so good, I might invite him to come and lecture to the undergraduates. So you've heard from the wonderful leaders of this organization, and I have to say it's one of my favorite audiences to speak to. I give talks to Harvard undergraduates and medical school students, but I always feel when I come to the JDRF that they want us to be successful, and we're in this together. So thank you all for coming tonight. And I'm going to talk in a different way from what you've just heard. You've heard a really nice overview from professionals about how to think strategically in a business way, how to organize this large organization to make it successful. My talk will be more or less the opposite. You might think of me like a scientific nerd focused on one thing. And so I'm going to bring you back to high school biology and talk about a simple-minded approach to try to cure the disease. So for those of you who don't know much about the disease, I'll give a brief introduction. And then I'm going to focus in on an experiment which is sort of an obvious one to do, but has taken our lab 10 to 15 years to achieve. At the end of that, I'm then going to spend a couple of slides talking about where I see the future going. But I warn you now, it's not such a big, broad vision. I don't think about the complications of the disease. I don't think about ways of providing better treatments on the margin. I want to get to the root cause and cure it. And so I'm going to tell you about one idea that I think can work. I might also say that, like the other speakers, I've been connected to the JDRF for a long time. And sometimes people come to me and say, I'm tired of hearing about there being, I forgot what the expression used to be, a cure around the corner or something like that. And what I think people don't realize is that science is not easy. It's hard, and you're not going to cure this disease in a month. By way of introduction, I'll maybe tell you a funny story. When we reported this finding recently, several of the reporters said to me, well, I understand what you've done. I don't understand why it took so long. And they kept going back and back saying, how could that possibly take 10 years? And it occurs to me that a newspaper reporter looking for something new every day, a businessman often looks for things on a quarterly basis. Scientists don't think that way. You have to think about the long term. So finally, I just told her, I don't know, maybe we're stupid and someone would have solved it much faster. But it did take us more than 10 years. So by way of introduction to finish, I want to thank the people who introduced the talk tonight. And I'll be joined soon by Julia Greenstein, who's been very helpful, and Kelly for organizing this meeting. So what I'm going to do then, as I said, is introduce the problem and then tell you about a potential solution. And the problem begins with the organ many of you will know, the pancreas, which is pictured here. It's about the size of a banana. It used to be thought centuries ago to simply be a cushion to stop your stomach from bouncing on your intestines, until in the 19th century it was removed and discovered that when dogs didn't have their pancreas, they were unable to digest food. And it was further discovered that they urinated a lot, which reminded people at the time of patients who had diabetes. In that pancreas, which as I said is about the size of a banana, if you go to fancy French restaurants, you might eat it sometimes. It's called sweetbreads in some restaurants. Inside that banana are tiny little islands or islets, which you've also heard of, pictured here, called islets of Langerhans. And I'm showing you one picture here. It's a confocal image of an islet. And your pancreas, if you're not diabetic, has about 500,000 of these little spheres peppered like raisins in raisin bread, let's say. What you see here in this picture are 4 or 5 different colored cells. They're stained for the hormones they produce. The green cells make glucagon, and the blue cells make insulin. And those of you who are diabetic and members of the family will know that it's the loss of the beta cells, shown here then in white, which enables or disables the person from making their own insulin and puts them on a necessary life support, you could say, of insulin injections. So I'm now going to give an unfair, kind of mean history of the last 100 years, namely, of diabetes research. And I don't mean to discount all of the wonderful things that have been done, but I would make the argument that essentially only one thing of any substance has been done, which is the discovery of insulin. So for insulin-dependent diabetics, injecting insulin has really been the solution since the 1920s. You might be amused here by this picture is a pile about 12 feet high of pig parts, which were used to isolate pancreases from pigs, and then they were purified through a complicated biochemical scheme to inject pig insulin. And that went on for several decades. There you see 2 tons of pig parts, which produced 8 ounces of pure pork insulin, enough for about 10,000 people for 1 year. And Eli Lilly was the company that really did very well at developing the biochemical purification of this. So for decades, I would say nothing much happened. And then in 1978, the gene encoding the insulin protein was cloned by Genentech. And that then led to these advances. Lilly produced the first human insulin, then they've made long and fast-acting insulin. Novo Nordisk really, in my view, didn't do anything other than invent a pen for injecting it as opposed to a syringe. Sanofi's made long-acting insulins, and we heard tonight about other versions of insulins which are coming along. And I don't want to say they're not important. They do improve the lives of people. But that's not, in my view, the right way to, it kind of nibbles at the problem. I want to go to the cause of the problem, which is that the beta cells are missing. So what I think about is another approach known to many of you, which is islet transplantation. So here's a picture given to me by my colleague Gordon Weir from the Joslin. And these are human islets isolated from a cadaver and stained with a chemical called dithazone that turns the insulin red. So you can see there are many of these spheres full of insulin. I don't know if we can see them here. And there's some contamination of islets or other cells that aren't making insulin. But this is really known to be extremely effective when transplanted. That controls a person's blood sugars far better than any insulin injection can, far better than any artificial pancreas does. It makes people euglycemic because the beta cell has 2 important properties, both of which are being replaced by some mechanical intervention. The first is it reads your blood sugar not every hour, which would be a lot for a diabetic, not every minute, but every millisecond. So think of like the thermometer in your house, reading the temperature inside every millisecond. The second thing it does is squirt out not the approximate amount of a long-acting insulin or short-acting insulin. It squirts out the exact right amount of insulin to keep your blood sugars under control. So the idea here is relatively simple. Let's replace the beta cell. Let's not use a mechanical device, but let's find a biological, I would even suggest a natural solution to the problem. So I worked for some years on something that turned out to be wrong, which was to try to find an adult stem cell that made the beta cell. Many of you will know, of course, that your blood system is topped up by a hematopoietic stem cell. When you go to give blood in a blood drive, you're not in permanent deficit for blood. Your body has a stem cell that makes more blood. And it had been believed that there was an adult stem cell that would make more beta cells. And it was in 2004 that we showed that that's not true for the beta cell. There is no adult stem cell. When your beta cells are all gone, your body has no ability to replenish it. There's nothing in the bone marrow. There are no other cells that become beta cells. I'm not going to show you how we did that, but it was a sort of complicated genetic lineage tracing experiment that showed that when new beta cells are formed, they are only formed from old beta cells. And so if you get rid of all the beta cells, there's no source of new ones. So that then led me to think about another approach, which was to use human embryonic stem cells, which can make any cell in the body. And that's where I happily first met John on the telephone. And I might take a little diversion here in a couple of ways. One is this picture just shows you from where we isolate human embryonic stem cells. They're from leftover material from in vitro fertilization clinics. But it also allows me to emphasize something that Derek and John said, which is the breadth of the JDRF in areas that people don't often know is extremely important. In my view, our nation teetered on the precipice of disallowing this kind of research. And what's not known to many people is that John and Leah Mullen and others at the JDRF arranged for people like me to see the president and others and have them rethink this idea of preventing this research. In the end, the compromise was still not what I would have liked. It slowed our research for at least 8 years, but fortunately it didn't stop it, and I thank John for his involvement in that. So this is just a picture, a cartoon of how we begin, that is where we derive the human embryonic stem cells. And this next picture is the one that really has occupied all of our time. The people in my lab make fun of this diagram. They call it my Skittle diagram. And it, in a sense, is the challenge that we faced. The red cell on the left, a human embryonic stem cell, can make any part of the body. So we had to figure out how to tell it to make the cell on the right. And the approach we took was to learn what I might call the molecular biography of a cell, from the fertilized egg to making a pancreatic beta cell. So imagine that we watched it at every stage of its development, removed it, and asked which genes did you turn on and off. All of your cells have about 20,000 genes. Any one cell has about 8 to 10,000 expressed at any one time. And so if you're a mathematician, you can do the math and see that that's an extremely complicated problem. You might think about it in this way. If I were to ask you who made you what or who you are now, you would think about the role your parents played, your teachers, your friends, the books you've read, the experiences you've had. That same thing is what we had to learn about how to make a beta cell. And the color diagram here shows one way we did it. We would take out genes that we knew to be turned on when cells were becoming, let's say, pancreas as opposed to muscle. Then we would manipulate the genes by genetic engineering so that when that gene came on, it made the cell literally fluorescent, might make it fluoresce green. Then we could purify those cells and ask what other genes are on then. That iterative process took us a very long time. We started it in frogs, we did a little bit in zebrafish, most of it in mice, and some with human fetal material. I think it's fair to say, with maybe 1 or 2 exceptions, we now know more about how to make a beta cell than any cell in your body. We know which genes come on and go off at practically every stage. There's a little bit more for us to do to get it completely finished, but we're very confident that we have a good idea of what genes need to be turned on and off to make the beta cell. If you look at this picture here, there are some terms that might not be familiar to you, like endoderm is the first step. The endoderm is the part of your body that makes all of the tissues and organs from your mouth to your anus. So if you like to do fishing, if you ever gut a fish, you're pretty much taking out all of the endoderm. So it's the mouth, the esophagus, the lung, the liver, the pancreas, the stomach, the intestines. That's all endoderm. So the first decision a cell had to make was to become endoderm, not to become mesoderm, bone, blood, or muscle, and not to become ectoderm, skin or nerve. Then we tell it to become pancreas, then endocrine, and then finally beta. And the beta cell here, an important part, is that we wanted it to become a beta cell that could sense glucose, that is GS for glucose sensing, and secrete insulin for IS, insulin secreting. So we weren't the only ones trying to do this. Many groups have tried to do this, many funded by the JDRF. And everyone was pretty much on the same pathway and got stuck at this pancreatic green stage where the cells can make all the different cells of the pancreas but couldn't get much further. And because of the exigencies, I guess you could say, for companies, they could not invest, I think, the time any longer to get to the end game. And so one of the companies mentioned tonight, ViaCyte, and another, Betalogics, decided to go ahead with a clinical trial with cells at the green stage. And how did we know we were on the right path? Well, we know because if we take cells at the green stage, here, cells that can make all the parts of the pancreas, and transplant them into a mouse that doesn't reject human tissue, 3 months later they will make some cells that are insulin-secreting beta cells. So they mature, as we would call it, by putting them into the kidney capsule of a mouse. As I've said then, 2 companies have decided to go ahead with a clinical trial, and I and others are cheering them on. As I think John nicely described, this is a really important first step to find out that things are, first of all, safe. While that device they're putting in will not make enough insulin to control a person's blood sugars, I'm hopeful that it will at least prove the principle that this approach can work. So we nevertheless went on to say, can we make mature beta cells? So that is a sort of good overview, and now I'm going to take the chance of showing you one really complicated slide. And forgive me for showing you this, but I have to because it's the result of so much work. So this is our solution to the problem. This is a solution where you see the different steps we've identified, and then the names there I'm not going to bore you with, but they stand for chemicals or proteins like keratinocyte growth factor, or this KIR molecule is an inhibitor of a protein called GSK3 beta in the Wnt pathway. And this would only be of some interest to molecular biologists, but this I'm showing you because it shows that unlike something simple like adding one drug, an antibiotic, we had to figure out the combination of things at each stage. This is 3 days, 3 days, 2 days, 5 days, 7 days, and then 7 to 14 in a 40-day process. And we had to figure it out so that it was reproducible. And I wish I could tell you that I had some really brilliant ideas for how to solve this problem. Most of my really good ideas turned out to be wrong, and much of the solution came from trial and error. It was an iterative process of guessing, going back to the beginning, and that is not a lot of fun. It takes people who are really dedicated, and I'm so grateful to the people on my team over a period of 10 years who've stuck with this problem. I'd say a couple more things about this. The second is that I don't believe this is a unique or the only solution, and we will work now with JDRF funds to improve it, to make it more efficient, to industrialize it, to make it more reproducible and better and faster. But I do think it's worth taking a big breath and saying at least this has cracked the back of the problem. Some people said, for example, you could never make a beta cell outside of the body. If we go back to this analogy of who you are, you had inductive interactions from your parents and your classmates and your teachers, other cells, you could say, telling you what to do. The problem here was we were doing this outside of the absence of any blood vessels or human serum or anything, so it was a pretty big risk to say, could you actually make a cell if it wasn't being made inside a living organism? The second thing we did from the beginning, and here again I would thank many of my colleagues, as John and Derek said, there are lots of people working on this, was we didn't want to just make a few cells. We didn't want to publish a paper. Someone once told me a funny expression I like is, no one was ever cured by by injecting themselves with a Nature paper. And so we set the goal of could we not only make these cells, but could we make them at a scale that would make a difference for people? And I'm going to show you this here. So this spinner flask on the upper left is about the size of like a blender, a food blender. And what you can see on the right is we figured out a way to grow these cells at scale. At 0 hours there, you can see some single embryonic stem cells. And they grow very quickly. They grow like weeds. 24 hours later, you see a sphere, and 3 days later, we now grow them in these spheres that are just the size of a human islet. This is one of the wonderful things about human embryonic stem cells is they grow so quickly, you can make a mass of cells very fast. One of the puzzles I give to the Harvard undergraduates, which I'll give you, I forgot to tell you, I'm going to give you a quiz at the end of this, is if you take a cell that's a half a micron across, which is smaller than the diameter of a hair from your head, and it divides once a day, and there's no cell death, what mass of cells will you have at the end of a year? So I'll let you calculate that, and then I'll tell you, I'll tell you the answer now. The answer is astounding. You will have 10 to the 47th Earths. In 3 months, you would make an entire Earth, because this is exponential growth. So there's a lot of cell death going on, but we can harness the power that these cells divide once a day, every day. So here we have them growing. Here they're being stained for some of these genes I mentioned. PDX stands for pancreatic duodenal homeobox. It's a gene which marks all of the cells of the pancreas, the duct cells, the exocrine cells, the endocrine cells, and here's C-peptide, a part of insulin. But I thought I would show you, if I can get this to work here, yeah, a nice little movie to give you a feeling for what this looks like. This movie, there you see a person's hand to give you an idea of the scale. So here are cells growing, at 2 hours, and then it'll just have a quick time course here. And the main point of what I want you to see here is in the last frame, which is that you can see little white specks there, these spheres growing. That incubator has 6 flasks in it, which is enough for 6 people. That is to say, we now grow enough beta cells in one of those flasks for 1 person. So we've solved the problem, as it were, at making the cell we want, and at a scale that is more than just looking at 1 or 2 cells. We can make billions of cells. So I'm going to show you some of the data that has convinced us and others that we've made this cell. Some of it's a little bit complicated, but I'm sure you'll follow it. This picture here may be the best way to show it, in that when a beta cell, remember I told you a beta cell makes insulin, but it also acts like a glucometer. It measures sugar. And so it gets rid of the need to prick your finger because it has sensors built into it called glucose transporters, which measure how much glucose is on the outside of the cell. So if we isolate from a human cadaver adult primary beta cells, and then in a dish challenge them with low and high glucose to ask whether when there's more sugar, do they secrete more insulin? That is, low is 2 millimolar and high is 20 millimolar. So this is one of the best human islet preps we've obtained. And you could think of this as like giving it 3 meals, breakfast, lunch, and dinner. We have low, high, low, high, low, high. In each case you see there's a spike in the insulin secreted. And then you have the cell dump all of its insulin by giving it potassium chloride. In the middle are the cells that ViaCyte and Betalogics are going to clinical trials with, which don't respond to glucose, but will, as I said, after incubated in a mouse for 3 months. And we all hope the same will be true after incubated in a human for 3 or 4 months. On the left are the cells we made, these new differentiations, low, high, low, high, low, high, and potassium. That, in effect, is the proof that what we've done is what we set out to do, is to make cells that could respond multiple times by sensing the glucose and secreting the right amount of insulin. Another assay or test for it is, can they cure a mouse of diabetes by transplanting them? This is a little bit complicated to look at, so I'll explain what you're seeing. This green color here and over here is not insulin, it's the background fluorescence. And in this swath of cells here, were transplanted the cells that ViaCyte has now put into human clinical trials. And some of you will be able to see a few green cells, there, 2 weeks after transplantation, and this is into a mouse. Here are our cells 2 weeks after, and you see 2 things. One is there are many more green cells, and there are also some red glucagon-producing cells, and these islet-like structures, which I'll show you here at a higher picture, look very nice. They're making abundant amounts of insulin and glucagon. So we can cure mice now very easily, but as my daughter says, she's sick of hearing about curing mice. So we've started some clinical trials, or sorry, not clinical trials, large animal trials, again with the help of JDRF, a JDRF-supported investigator at MIT, Dan Anderson, and in collaboration for the monkeys with Jose Oberholzer at Chicago. And here I'll show you just the beginning of our work together. These are islet-like structures in a monkey after transplantation. And a nice sort of anecdote here is that when Jose showed it to the pathologist says, yeah, I've seen a lot of these human islets from cadavers you've transplanted before. What's new? Well, what he didn't know is what's new here is these have been made from a human embryonic stem cell, and unlike human cadavers, are now available in unlimited quantities. So I'm going to end with a few puzzles and then point to where we're headed. So the puzzle, you could say, so all right, what would you do with an unlimited supply of human beta cells? There's a picture of human beta cells made from human embryonic stem cells, insulin stained in green. Some of you might be able to say a few red cells there. Those are glucagon-expressing cells. In the black regions inside the sphere are cells that are making neither insulin nor glucagon. And I'll come back to those in a moment. So for me, what you would do with it is sort of obvious. But for others, for example, the pharmaceutical industry is keenly interested in using these cells to screen for drugs to treat type 2 diabetics, to prevent beta cell dysfunction, to improve beta cell secretion. But I'm really focused on this. And forgive this simple cartoon. I actually am a very simple-minded person. In fact, I'm reminded sometimes when I submit grants, they're rejected because it says they're overly ambitious and simple-minded. I actually am flattered by that. I like very simple things. So here's a diabetic which is not able to control their blood sugars. And the idea is to take a human stem cell, turn it into a beta cell, protect it, and make a normal person that doesn't have to prick their finger to read their blood sugar, doesn't have to inject insulin, can eat whatever they want, and can play soccer whenever they want. That's my simple-minded goal. I think we're very close to that. I don't want to tell you we've solved the problem, but we are so close to the goal line. Essentially, we are at the goal line because we've broken the back of the problem. We've shown that we can make these cells. I now want to spend a little bit of time telling you about the challenges that we face going forward in the next year, and then I want to look out a little bit farther and have you kind of dream with me about what the perfect solution would be. So one of the things that concerns me now is shown here. This is a different picture of one of those clusters. It's been fixed and then sectioned like slicing salami, this sphere. So you're looking at a thin section through it. And the cells that are making insulin are stained brown. These cells here are obviously not making insulin. A few of those are making glucagon, which is a good thing. Most of them just haven't got with the program. They might be stuck progenitors. They might differentiate 3 or 4 months after transplantation. We don't know, and I want to figure out what those cells are. Should we remove them before transplantation? Should we give them more time to differentiate? That's one of the challenges we'll try to solve this year. Related to that is I see no reason not to try to make an islet. We focused on the beta cell because the beta cell is the cell missing in diabetics. That's what I've talked to you about today, this purple cell at the top. So we begin with the stem cell, we treat it, and we can make these clusters that contain beta cells. I also mentioned to you that the clusters contain a small percentage of alpha or glucagon producing cells. Well, very recently, Quinn Peterson in the lab, with the help of Nairi Brown and others, has been working on how to make these other cell types. And Quinn has now got it so that he can make a cluster that's mostly alpha cells and not many beta cells. Moreover, he's confident he can make this delta cell, which makes somatostatin. And so I think if I'm invited back in a few years, I'll be able to tell you that we can now reconstruct an islet. You can tell me what portions of what cells you want, and we'll dial them in and make a personalized islet, whatever you want in terms of these kinds of cell types. In fact, this last one we're not really worried about. As far as I know, diabetics don't have trouble with appetite, and this controls appetite. This is an interesting one, pancreatic polypeptide. Even though it's been known for decades, no one has a clue what it really does. But we can nevertheless at least make this one, and this one already. So one of our challenges then is to get better control over this process, to, as I've said, not only treat with chemical combinations, but reduce the time and the cost. Another way of just saying improve the efficiency of the process. To give you some idea for it now is that when we make the cells in that flask, it takes us 30 to 40 days. It works more than half of the time, but it doesn't work all the time. We have to tweak it to figure out what the problems are. And each flask costs $6,000 just in the cooking material, just in the rest, the parts of the recipe. So it's an expensive process. I'm confident we can reduce that, make it more efficient, but that's going to take us time this year. So in the next year, that's what I'm really going to focus on. But I now want to come to this other business with blocking the immune system. As John nicely said, there are lots of immunologists who have been thinking about this problem, and there are various approaches to it. And I'm cheering them on, but I don't like any of those approaches. Most all of those approaches involve trying to diminish or reduce the severity of the immune attack. They are general immunosuppressants, or some are even more specific than others. I have a different idea, which I'll explain in the next slide, and to tell you about what I'd like to do. So in order to protect the cells, we really just need them to let glucose go in and hormones come out. Here you show a picture, I show you a picture of encapsulated cells, encapsulated with alginate from our colleague Dan Anderson at MIT. And the idea there is that immune cells are trying to attack these, and this encapsulation method might protect them. Like with the artificial pancreas, I think encapsulation is likely to be the first thing that goes to market. I don't know in what form, but I think that that's likely to be the first way these cells get into people. At the same time, I don't believe that's the best solution, because those cells cannot have an intimate contact with blood vessels, therefore they won't be able to read blood sugar as perfectly. Plus, those capsules will eventually degrade, and there aren't enough nutrients that go in and out to keep the capsules and the beta cells inside of them happy forever. And I want a forever solution. So what I want to do is start on encapsulation, but work on biological protection. And so I'll finish by just having you think with me about how you might protect the cells. And I'll tell you two of my favorite ideas, but remind you that most of my ideas turn out to be wrong. The first is that cancer cells do what we want these transplanted beta cells to do. Many tumor cells have figured out a way to block the immune attack. Immune cells go to the cancer cell and try to kill it, but the cancer cell is quite clever. It expresses a ligand on it called PD-L1, protection of death ligand 1, which essentially is saying to the immune system, leave me alone. There's a complication in the immune system that it usually requires co-stimulation, two signals, and this blocks one of them, so that the T cell, which is going to kill the cancer cell, just leaves it alone. So one idea would be to make use of what cancer cells have selected for and express PD-L1 in our beta cells. Now the FDA will say, whoa, that sounds like a bad idea. You're turning the beta cell into a cancer cell. But the big difference is cancer cells divide and these beta cells don't. So one thing we're going to try in the next year is to express PD-L1 and another blocking protein called CTLA-4Ig and see whether or not we can stop an immune attack. My favorite idea, though, is a different one, and this has to do with one of the most basic facts of life, which has always been a puzzle. And that is, let me think of a good way to explain this. When a mother has a baby inside of her, she should, given what we know about the immune system, reject that baby. And the reason she should reject it is if the father, let's call him the sperm donor, were to have given a skin transplant to the mother would reject the father's skin because it's not hers. The fetus is half of the father, so why doesn't a mother reject a fetus? I'm not the first person to wonder about this, but no one has actually figured that out. So my idea is that special cells around the embryo, called trophoblast cells, are expressing ligands on their surface, proteins on their surface, which tell the mother, even though I'm not you, leave me alone. So another way to think about this is to express those ligands on our beta cells and have them look like fetal cells. So my dream is to come back sometime and tell you not only can we make billions of beta cells, but we can transplant them into any person and they won't be rejected. I don't know that we'll achieve that, but I know that we wouldn't even come close were it not for the JDRF. I'm going to finish by making note of the people who are working in my lab now on this. They're listed here in the left in white, some of our collaborators in yellow on the right. And it might just be worth mentioning that a lab like mine spends a lot of time, as John and Sean and Derek all said, raising funds to do this kind of work. In my case, we have at any one time about 13 or 14 different grants. I'm funded by the Howard Hughes Medical Institute, the Harvard Stem Cell Institute, the National Institutes of Health, the JDRF, the Helmsley Charitable Trust, the Pickauer Foundation. I'm very grateful to all of them, largely because they have continued and sort of stuck with this over years when it wasn't so clear that we would be successful. So I'll be glad to take questions, but I want to end really by thanking all of you. And I say that because people talk about their children, and many of you are here have done a lot of volunteer work. I don't do any of that sort of stuff. I'm not good at business or strategies or things like that. I like being in the lab, and I want to solve this problem. But I wouldn't be able to do it if it wasn't for us all working together. I don't want to sound corny like we're a family. We're kind of like a dysfunctional family. But it's really helpful to have you all joining in this process of trying to cure this disease. So thank you.